Hi, everyone. Thank you so much for joining us, both in person and virtually, to see Rachel Crane give our fellowship is a fellowship that's funded by the NSF and administered through the American Society for Engineering Education. And through that fellowship, she's currently working on mechanics of the dragonfly. But before all of that, Rachel started her educational career at Swarthmore College, where she studied mechanics of worms, on to be a manager in the Sheila Patterson Lab at University, where she studied mantis adaptation. And then she went on to her Ph.D. at Stanford's Space Station. So she's going to be telling us today about her work on where she studied resistance and repair in mussels. But I will also add that she was my roommate and my best friend through grad school, and so clearly we have to address that here as well. So it's really exciting to see your friends like become incredible scientists and to be able to grow as a person and a scientist with them. And so I'm really, really happy to be able to share my friend and colleague Rachel with you today. Thanks.
All right. Thank you, Elora, so much for that introduction and for the invitation to talk to you all. It's really a delight and an honor to be here. And I'm so excited to talk to you about bivalve shells, this really amazing biological armor. So these are all bivalves. So bivalves are a type of mollusk, includes animals like mussels, oysters, clams, and scallops that have a shell composed of two valves that are made of this really brittle material, calcium carbonate. Brittle meaning it fractures really easily, and yet with just a little bit of organic matrix, these shells become this really effective armor. So I want to start with a question. How do we measure how effective an armor is? Like, how do we meaningfully know how good something is at protecting the animal inside? And I'm going to start with a little bit of a goofy example. Okay, so let's say you take up jousting. You're going to want to buy armor that can protect you from getting hit with a lance. And you're going to want to make sure that that armor is good enough to make sure that you are not harmed or injured at all. And so one thing that you might be interested in is the strength of your armor. And strength is a measurement of the force required to break a structure. And a force is just like a push or a pull on an object or a material. It has a magnitude and a direction. And so you want to make sure that the strength of your armor, the force that's required to break it, is larger than the force of getting hit with a lance. And then you know that if you get hit with a lance, your armor's not going to break and you're going to be great. And we can do this with shells. I've done this a lot. Lots of people have done this. So this is a mussel shell, and I'm crushing the shell. I'm imposing a compressive force and measuring the force required to break the shell. So that's great. You buy your armor, get hit with a lance, and you're perfectly fine. But if you are into jousting, like you're probably going to joust more than once, and you're probably going to get hit with a lance a lot. So like get hit 10 times, 50 times, 100 times. And it's really very possible that you're going to get hit with a lance 99 times and nothing's going to happen. But that 100th time you get hit with a lance, even if it's exactly the same magnitude force, your armor might break. And if it does, it's because of mechanical fatigue. So fatigue is a process where repeatedly applied forces, in this case, getting hit with a lance, causes accumulating damage. So there might be all sorts of microscopic damage to your armor that you can't even see that then weakens the structure. So your armor is less strong. So a lower magnitude force is sufficient to break your armor. And this is a huge problem, right? Like this is important in any sort of design or manufacturing or civil engineering, right? Anything we build, we need it to be able to withstand not just really, really large magnitude rare occurrences, but also the forces associated with daily use, which means this is also important in any biological material. And y'all might be familiar with this. So We're all vertebrates. We all have bones, and maybe some of you have broken a bone before. Like maybe when you were a kid, you fell off the monkey bars and you broke your arm. And if you did, it has to do with the strength of your bone. The force of hitting the ground exceeded the strength of your arm, so your arm bone fractured. Some of you might be familiar with stress fractures, though, as well. These are actually associated with fatigue. So stress fractures occur when lower magnitude repeated stresses, like maybe you started training for a marathon and started running a whole bunch really suddenly. That repeated impact of running and your feet hitting the concrete can cause stress fractures, which is damage that accumulates in your bones due to fatigue. So what I'm going to do today is I'm going to use this perspective of thinking about mechanical fatigue, of thinking about lower magnitude forces that animals have to contend with on a daily basis to try to better understand bivalve shells as an armor. And I'm going to use the California mussel here on the California Rocky Coast as my model system. And these animals, these armors have to defend against all different kinds of threats. So there are predators that'll swallow them whole, that'll drill holes through their shells, that'll pry them open. Perhaps more relevant if we're thinking about fracture, there are animals that will hammer shells to break them, that will crush or peel them open. And then birds will also just drop shells from the sky so they shatter on rocks and concrete below. In addition to predatory threats, there are environmental threats. So wave impacts, like those huge storms that we just had, both the storm directly impacting animals as well as throwing debris, rocks, logs, whatever at the intertidal zone can cause large amounts of damage to the animals that are there. And then, because shells are a living animal, there's this really interesting wrinkle that gets thrown in when we start to think about survival in response to accumulating damage, and that's repair. So if you take up jousting, your armor is not a part of you. It's something external that you put on and you take off when you're done. Bivalves are, of course, living animals. They build their shells themselves. They deposit the calcium carbonate that makes up the shell, and they can repair from many different kinds of damage. They have really plastic shell forms in a lot of contexts. And so if we think about armors as something that has to respond to accumulating damage, the animal's capacity to repair could also have a large effect on how good that armor is. So returning to bivalve shells as armor, I'm going to tell you about a series of 3 experiments that I've done looking at mussel shells through the perspective of mechanical fatigue. So the first experiment, we're just going to talk about whether mussel shells experience mechanical fatigue. I'm then going to look at whether or not they can repair the types of damage associated with fatigue. And then finally, the whole premise of fatigue is that we're looking across long durations. So we're going to look at their repair response to a chronic mechanical stress. Before I get started, well, I've already started, but before we talk about the experiments, I'm going to introduce you to my study system. So this is my field site. This is at the southern edge of the Monterey Bay. We're looking out across the Monterey Bay. This is my field site, and it's in the intertidal zone. So if we wait for the tide to go out, we can walk out, and there's this huge expanse of life there, including these mussel beds. So California mussels live packed in cheek and jowl with their neighbors. They're super easy to collect. All of my experiments were done on relatively small but adult sexually mature mussels. And because I'm interested in damage, unless I tell you otherwise, I started with animals that had little to no apparent external damage. So just to try to start with as clean a slate as possible. Cool. So, do mussel shells experience mechanical fatigue? I specifically was asking whether shells can be broken by chronic subcritical loads, so something that's below that strength, that one-time breaking force. If those loads are applied cyclically, meaning you apply the force and you remove it and you apply it and remove it and you do that over and over and over again, which is a classic mechanical test of fatigue, or statically, where you just put the force on and you leave it there and you see what happens. So cyclic loading, this is the same device I showed you earlier. So I took one valve of a mussel shell and I loaded it to a predetermined force. I did this all in a saltwater bath. And I just did this over and over and over again, and somewhere between 10 and 100 and 100,000 force applications, my mussel shells broke. So I know what force I was applying, and I know how many cycles it took the shell to break. And I'm going to plot those data here. I'm going to take this with me. Okay, so this is a little bit of an unintuitive figure. It's a variation of a traditional engineering figure. So on the x-axis, this is my number of cycles. So how many times it took the shell to break. And this is the force that I was applying proportional to the predicted strength of the shell. So I divided the force that I applied relative to how much I was expecting the shell to be able to withstand. And then that line at 1 means that the force I was applying was equal to the strength of the shell. Anything above that was a higher force, and below that is lower. And then note these are both log scaled. Okay, so here are my data. And as you can see, there's a really strong negative relationship, which means these shells are experiencing mechanical fatigue. A lower magnitude force is sufficient to break a shell if you apply it over and over again. And this effect can be really dramatic. So this is 50% of the predicted strength. That's a huge decrease in the force that you're applying. And that's enough to break a shell within only 1,000 cycles, which is not that much if you think across the lifetime of one of these animals. And especially because of these log scales, you can have really dramatic effects of mechanical fatigue in terms of how resilient these armors are. We also looked at static fatigue. So I built this apparatus I can run 8 shells at the same time. It's exactly the same loading. So in salt water, it's a single valve from a mussel, and I hang a force off the end of this lever arm that just applies a constant force to the shell, and then there's a switch that triggers when the shell breaks, so I can time how long it takes the shell to break. So we just load the shell, see what happens. This is the same figure I just showed you with force proportional to strength on the y-axis, and x-axis is now duration in hours, and they still break. Like, you don't need to cycle, you can just leave it there, and eventually the shell breaks. It can take a really long time. This is my favorite data point I've ever collected in my whole life. This is 409 days. So for over a year, that force just sat there on that shell, not doing anything as far as I could tell. And then one day I came in and the shell had broken. So, and as we stretch out this time scale, you can see that the forces that we're applying are really, really low relative to the strength of the shell. So I have breakage under both cyclic and static loading, and I was curious how these two curves compare to each other. You can actually directly convert cycle duration to a normal duration by integrating under the curve, and I did that, and we plot these two figures on top of each other, and the slopes are exactly the same. So there's nothing special about that loading and unloading. All that matters is how long the shell is loaded for. This is really surprising. I'm going to tell you guys right now, this property that cycling is not important is not true for all sorts of materials, both manufactured and biological materials. Sometimes it's true, sometimes it's not. I was really surprised because we're looking at this complex structure with multiple materials layered on top of each other in this complex shape, so that we're still finding this consistency of this result is really surprising, and it's really helpful from a few perspectives. So it means that experimentally, we can fill in a lot of, we can test a lot of different kinds of forces and durations. These two experiments are good at different things, and so we can fill in a lot of the space to better understand how different forces contribute to shell breakage. And it's also really cool ecologically, because it means we can look across all different kinds of forces that animals have to contend with to understand how they contribute to damage accumulation in mussel shells. So I'm going to do that. Okay, so this is the slope that I just showed you. I've now put it in terms of just an absolute force instead of that proportion, so I can compare it to actual forces that mussels encounter in the wild. So this is the bite force from an otter jaw, and you can see that it is well above this line. Otters are going to have no problem cracking a shell like this with their jaw. A crab, on the other hand, is a typical large crab from our area. You see it's unlikely to be able to generate enough force to break a shell outright. But if it works at a shell, it might be able to. And if you hook up a force transducer to a crab claw as it's handling a shell, it does just that. It works at the shells, and they'll actually pulse while they're holding onto the shell. So they are applying repeated forces as they're trying to crack open shells. Something like a horn shark is not going to be able to break through these shells. A super rough back of the envelope calculation for drag imposed from a wave force, that's going to have to be continuously applied for 66,000 years. So probably not going to be relevant, we can confidently say, but there are other threats so that the impact is much harder to calculate directly from the wave, as well as those wave hurled debris could still be really important in terms of contributing to damage accumulation. I also think it's the effects of climate change could be really interesting here, and ocean acidification often has, there's been lots of testing on how ocean acidification affects the strength and repair of calcium carbonate animals, but we don't know how that affects the slope of this curve. So whether it just moves the curve or shifts the slope could have really profound effects on how the animal contends with daily stresses. Okay, so summarize this section. Mussel shells absolutely can be broken by chronic mechanical stresses, whether they're applied cyclically or statically, and this sort of damage accumulation occurs on biologically relevant timescales. Okay, so these shells are potentially accumulating damage. Are they able to repair and respond to this? So we know that mussels and lots of bivalves generally have a really remarkable capacity for repair. So this is a mussel shell that my lab mate brought me because she thought I would like it, and she was correct. And there's a big hole in the outside of this mussel shell. You see this all the time. And what you see through that hole is not the ooey gooey insides of the mussel. That's actually new shell that the animal has deposited to patch over this hole. So this animal experienced sort of catastrophic damage, but was able to patch up that hole so that it survived. But we don't know whether mussels are able to respond to the types of damage that are associated with fatigue and repeated loading. So that's sort of question one. Can they respond to that? What is the time frame for that repair? So how long does it take them? And can we see visual evidence of repair? Can we see what's happening inside the mussels? So for this study, I collected a whole bunch of live California mussels. I divided them into two groups, and we've got one experimental fatigue treatment group. They're going to be in this pinky peach color for the rest of this section, and I've got a control group in gray. And the fatigued mussels, I loaded whole live animals because I want to look at repairs. The animals have to be alive for 15 cycles at 67% of the predicted strength, which was enough for about half of my shells to break, which this was good. I wanted some treatment where like would have some animals survive, but it was enough loading that I knew I was doing something. And those half that died, that broke, I excluded. And then the remaining shells, remaining animals, I kept in the lab for 4 weeks with control animals. And then at a few intervals across those 4 weeks, I removed a subset of animals and killed the animal, dissected the shell, and tested the strength by loading the shell until failure. And then just to be really clear, this is not longitudinal because I'm killing the animals at each time point. So these are different animals. And so I'm going to show you our 4 time points immediately after treatment, and then 1, 2, and 4 weeks later. So this on the y-axis is the shell strength in newtons, and then I'm showing that relative to the shell mass, so a proxy for the animal size. And you can see that immediately after my fatigue loading treatment, the fatigued mussels are weaker than the control shells. They're breaking at a lower strength or a lower force, which that's great. That makes sense. We're expecting this. We loaded them. They should be weaker. They are weaker. That's awesome. 1 week later, there's no significant difference between my two groups. So this was wildly faster than I was expecting. I thought this experiment was going to run for 2 months. And within 1 week, there on average was no difference in the strength between my two groups. And this was maintained 2 and 4 weeks later. Something interesting also happened if we look in a little bit more detail just at the animals just at the fatigued mussels. And specifically, if I look at sort of how much force they experienced for their size. So I tried to load all the mussels to about the same amount, but there was variation around that. So for their size, some mussels experienced more force and some experienced less force. So what I'm showing you here on the y-axis is still the shell strength. And then the x-axis is what I'm calling the fatigue force residual, which is just if I plot a regression through how much I loaded each mussel for its size, this is how different that mussel was from that regression. So was this mussel loaded a lot for its size or not a lot for its size? So bigger forces to the right, smaller forces to the left. And immediately after the fatigue treatment, there's no effect of how large that force was on my measured strength. And that is still true 1 and 2 weeks after the fatigue loading treatment. But our 4th week, a pretty strong linear relationship emerges where shells that were loaded relatively more for their size are stronger than mussels that were not. If you are looking at these data right now, you're wondering about this point. It does have a lot of effect on this, but if I remove it from the data, that slope is still highly significant and is actually stronger than it was otherwise. So this is indicating that not only are mussels repairing within 1 week, but a month later, mussels that experienced relatively more loading for their size are stronger than mussels that experienced less loading for their size. Okay, so we've got fast repair, we've got this bolstering strength, but like what's actually going on inside of the mussels? So in addition to testing strength, I set aside a group of mussels, dissected them, and I looked at the internal surface of the shell with a few different kinds of microscopy. So first, just looking with a light microscope. On the left, I've got a little schematic of a mussel valve that corresponds to what this picture is. So we're not that zoomed in. This is most of the internal surface of a mussel shell. And this is a group that was fatigue loaded. And you will notice there is this massive web of fractures spreading across the internal surface of the shell. So there's a lot of damage on this shell. And we found fractures like this in about a third of our fatigue loaded shells and in one. And in one of our control shells. So this type of damage does occur occurs in the wild, but does seem associated with our fatigue treatment. These fractures could look really different. So this is an extreme example. Here's a shell that just had a little bit of fracture around the posterior lip. And sometimes this animal has, you can see there is fracture, but it's much smaller as opposed to this big web. There's just these 2 small lines of fracture at the center of the dome. So what about repair? So this animal that we've been looking at for a little bit has, we noticed these brown depositions around the locations where there were fractures. We didn't see this brown growth ever not associated with fractures, but we did see it in some of our shells around where we saw breakage had occurred. And we don't know exactly what this is, but for many mussels, the first step of repair, if you like drill a hole in the shell, is that they will cover that shell with periostracum, which is the brown outer proteinaceous layer of the shell. So that could be what's happening here. We don't know it, but it's an option. We also used calcine staining to look at our shells. So calcine is a fluorescent dye that we kept our mussels for a week in a calcine bath. And calcine binds the calcium carbonate that's in shells. So any shell that gets deposited during the week that the mussel's in the bath will be stained and can be visualized with the fluorescence. So we can see shell that's been laid down since the fatiguing event. And so here you can see, maybe with this level of light, but there is an increase in fluorescence, especially around the areas of fracture. And I'm going to zoom in, which the fluorescence should be brighter here. Okay, so you can see we have calcine staining not so much directly on top of the fractures, but in the areas surrounding them, which sometimes repair of like drilled holes again, which is most of what research has been done, moves sort of distally to proximally to cover holes. So this does seem to be matching that pattern. Okay, so we saw these 2 patterns in this shell. We did not see them in all of our shells though. So among shells that had been fractured, we saw that brown growth in about half of them. And we saw new shell deposition as evidenced with the calcine staining in about half of them. Actually, exactly half of them. Here's an example when that didn't happen. So we've already seen the shell. It's got those two simple fractures. You can see clearly there's nothing brown going around that. And if we look at the calcine staining, there's nothing there. The animal's actually busy depositing shell like elsewhere on the valve, not on top of where the fractures are. So mussels, in summary, are able to rapidly repair damage from fatigue. This mitigates the threat posed by fatigue because that repair can occur so rapidly. And we showed that fatigue loading is associated with an increase in fractures across the interior surface of the shell. And we saw in some of our shells evidence of a repair process with this brown growth and the increase in shell deposition, but we didn't see that in all of our animals. So we don't really know what's driving that rapid repair response when we look at our strength measurements. Okay, so this study was looking at repair and response to a single damaging incident. But of course, the whole idea of fatigue is that damage is accumulating across the animal's lifetime. So can the animals maintain this repair response across long durations? And more specifically, we were asking how mussels contend with a lifetime of accumulating sublethal damage. And for this experiment, we asked a series of questions. When faced with chronic cyclic loading, we wanted to know if mussels, first of all, will maintain their shell strength, so similar to the last study, but in response to chronic loading. Whether we'll see evidence of internal repair, so looking across a longer duration, we may better be able to see what that repair response looks like. We also wanted to look for changes in shell morphology. So a lot of mollusks have pretty plastic shell forms that can change in response to environmental cues. This has been well studied in response to chemosensory stuff. So like animals that get a chemical cue indicating there's a predator present might thicken their shells. And we were curious if this sort of mechanical stimulus can trigger the same response. And then we also were curious if there's a cost. If the animal's constantly having to repair its shell, is there some cost in terms of reproduction or survival? Okay, so once again, we started with a whole bunch of mussels, divided them into two groups, and we've got a fatigue loading treatment that here is in red and a control group in gray. We kept all of the animals in the lab for about 7 months, during which almost every week we took the fatigue loading group and we loaded them for 15 cycles at 55% of the predicted strength, which is enough for about a third of the shells to break. We measured shell morphology at the beginning and end of the experiment, so we could look at growth, look at changes in morphology. At the end of the 7 months, we dissected the animals and dried and weighed the internal tissue, and we separated out the gonads so that we could separately look at the gonad tissue and the non-gonad tissue. We visually inspected the shells to look for evidence of repair, and we measured shell strength. So first, starting with strength, here we're looking at strength relative to shell mass between our two groups, and I've plotted two lines here, but these two lines are not significantly different. So despite 7 months of chronic loading, that was enough to break a third of the animals. There's no difference in strength at the end of 7 months. So these animals are able to maintain their strength. To look at internal repair, we sort of visually categorized the extent of the internal surface area that was covered with a repair patch. And what I mean by a repair patch, so this is a non-patched mussel. The internal surface of mussels is, the calcium carbonate is laid down in this brick-like structure called nacre, which is mother of pearl. It has that beautiful opalescent, very distinctive property. And animals that have external damage will patch internally with a different type of shell that is sometimes a different color and is noticeably matte, does not have that pearlescent property, and it has these clearly defined borders, so it's really easy to see where the patches are. So we just, here are the patches. We just approximated the extent of the internal surface area that was covered with a patch. And so here's what we found. Our control group generally had no internal repair, and some of them had a little bit. whereas our fatigued mussels often had at least a small amount of internal patching. I was curious how this compared to mussels from the field. So, because remember I told you I started with animals that had no damage. So I went out and I just collected a whole bunch of mussels without that pre-filtering step. And this is what the amount of internal damage looks like. So just mussels experiencing normal field associated damage usually do lots of this patching, and actually, if anything, more than our fatigue treated shells. So what we're seeing is a very typical ecologically relevant response. What about changes in morphology? So are we getting plastic changes, differences between our two groups? Sometimes. So I'm going to tell you right off the bat, there was no difference between our two groups if we look at growth, changes in mass, differences in shell length, and differences in shell height. However, there was a difference in the growth along the width dimension. So here is the growth in width in millimeters, and you can see that our control shells got a little bit wider than our fatigued shells. This surprised me. I would have expected if shells were making any change, they would be changing in a way that would make them better able to resist the kinds of threats that they're encountering. And this measurement of width, it kind of makes the shell more domed, more rounded, which makes it stronger. So in all of our previous tests, wider shells tend to perform better. They tend to be stronger and more fatigue resistant than shells that are less domed. So this is the opposite of what I was expecting to see, but I think I know what's happening. So we also measured shell thickness at the sort of peak of the dome. So how thick is the shell at its sort of highest domed point? And you can see that our fatigued mussels, so the thickness here is on the y-axis and the shell size is on the x-axis, and our fatigued mussels are significantly thicker than our control mussels are. The reason why there are 2 points is there are 2 valves for every mussel. So I've just connected the 2 valves, but shown them independently. So what I think is happening is these mussels, they're all laying down shell, but instead of depositing and getting wider, our fatigued mussels are using that shell to patch and to thicken. Okay, so finally, what about a cost? So these animals, they're responding to this damage, they're depositing their shell a little bit differently. Does this come at a cost? So remember, I measured this by dissecting our animals and looking at the dried mass of the non-gonad and the gonad tissue. I found exactly the same thing with both, so I'm just going to show you the non-gonad tissue today. Because all of our animals were kept in the lab for 7 months, I wanted to compare them. I wanted a baseline. So these are mussels collected straight. This is, day 1 of the experiment. I took a group of mussels and dissected them and looked at the dried mass of the tissue. And you can see, unsurprisingly, it's correlated with shell mass. So larger mussels have more internal tissue. This is my control group. So our mussels did not like being in the lab. We tried really hard. We fed them a lot. But clearly, either we were not feeding them enough or they were just really unhappy. But there was a huge cost to being in the lab in our specific setup. There is a very slight but significant difference between the control and fatigued groups, though. So I think there's a little bit of a floor effect. It's hard to do worse than our control mussels were, but the fatigued mussels were slightly worse off than the control group. Another bit of evidence in terms of the cost is in the final few weeks of the experiment, likely having to do with the huge decrease in tissue mass. I found some mussels dead each week in the tank. So there was no obvious cause of death. There wasn't like a predator that had gotten in or anything. These were just dead shells with the animal dead inside. And this happened to 11% of our control mussels and almost 30% of our fatigued mussels. So these are significantly different. So there was a high cost to being in the lab, but that was exacerbated for the animals that were experiencing this fatigue loading treatment. So when faced with chronic cyclic loading, mussels are able to maintain their strength. They do repair internally by patching. We documented some changes in shell morphology. So the mussels that experience loading are less wide, but they tend to be thicker than their counterparts. And the animals that experienced chronic loading, they lost more tissue mass and were more likely to die than animals that had not. So coming in summary of everything. So mussel shells absolutely experience mechanical fatigue. Repeated forces will weaken shells. However, the threat that poses across these animals' lifetime is mitigated by their extremely rapid repair response. And that repair can be maintained across long durations and can be associated with changes in shell morphology, although it can come at a cost. So I started today by asking us to think about what makes a good and effective armor. How do we think about that? And by taking a slightly different perspective than the traditional one, by thinking beyond just shell strength, we talked about this sort of much more complicated and nuanced perspective of armors as this biological armors as this dynamic system that is accumulating damage, and then because it's part of a living animal, that animal has the capacity to repair and respond. So it's a much more complex perspective that's grounded in the ecology of what these animals are actually experiencing. I want to note that although I think I learned a lot by thinking about mechanical fatigue with a biological structure, fatigue is really understudied kind of broadly, but especially in biomaterials. There's very, very few biological materials about which we know anything about their fatigue properties. So I think there's a lot more to learn there. And there's also just so much left to learn about fatigue and repair and the mechanisms of both of those, both in mussel shells and in bivalve shells more broadly, about how these animals take this brittle component and create these really impressive armors. So with that, I want to thank my thesis advisor, Mark Denny, who worked with me on all of this, and a phenomenal undergrad who spent a bunch of time crushing and rearing mussels with me, all of my lab mates, and a lot of people who helped me, especially with the microscopy, as well as building and maintaining all sorts of little apparatuses and contraptions that I use throughout this study. Yeah, and thank my funding sources. And I will take any questions.
Thank you for turning that on. Hi, thanks so much for the talk. That was great. I want to preface my question by admitting that I'm a botanist and know absolutely nothing about any of this. But I was, you talked about all of these measures with a particular species that has a particular life history, I guess you would say, right? It's sessile. Do you use that term? Good. Do you have any thoughts or ideas about these types of patterns in mussels or in bivalves that have different life histories or that are not sessile?
Yeah, I would. You are describing the the over optimistic second year PhD student who writes an entire career's worth of experiments that are going to happen during their dissertation for their proposal. And I really, really was hoping to sample more broadly and to look across species. I think especially looking at different aspects of life history, what the shells are being used for, animals that encounter different levels of predation, animals that are in faunal and using their shells really differently to engage with the sediment. I think understanding the relationships between morphology and mechanics is really interesting there. And then way, do I have a slope that I can pull up quickly? The answer is no, then I won't worry about it. Yeah, here we go. Okay, so this curve, this was one of my things I thought a lot about. So usually when people measure mechanical properties, either they're doing nano indentation, which is really important, but does something very different. But usually you're just looking at this point. And so I have no idea as you look at different shells, if this slope of this line stays the same. And right, like if that slope is changing, that's going to have huge effects. And if it's staying the same, why is it staying the same? Like, I think that's really interesting. And I have no idea, but I think it'd be really cool to find out.
My question is about this graph because I really like this graph. I thought it was a cool way to show like practical things that could be occurring to these mussels. It made me also just think about like, well, was your area that you sampled from like a high traffic area of people? And if so, like where does like a human stomping like fall into this range?
Where a human stomping is a great question. So we were not in a high traffic area. My sampling was at Hopkins Marine Station, which is on an MPA. So it was on those grounds. So no one's there who's not a researcher. And I know there was no one doing like I was the only person who was out there. If I had the relationship between newtons and pounds better in my head, I could figure this out. But if anyone looks up how kilograms is, you could figure it out with weight. That I don't have those conversions off the top of my head. But if you step on a mussel shell, this type of mussel, you're not going to break it. I had another set of thoughts. Oh, yes. In order to get shells that had relatively little damage, I didn't talk about this at all, but if we're in the intertidal zone, mussels are found across a huge gradient. So these animals are really high in the intertidal zone, and then you can go down much lower and there are still mussels. The animals that are higher in the intertidal zone tend to have way more damage to them. I think a lot of that is actually not associated with predation. It has to do with, I don't actually know, but there's lots of things will like grow on shells and affect the wear more than, so I think it's more wear, but these higher, I may be wrong. If anyone knows, please tell me. But the animals higher in the intertidal zone have really different shell properties and different mechanical properties than the animals lower in the intertidal zone. And all the work that I talked about today was with animals low in the intertidal zone. I've done some morphology stuff about that gradient, but not a lot of it. But again, going with how, like, yeah, ecosystem has a huge effect on what shell form looks like. These guys are really different than the ones that I was sampling lower down. I rambled. Did I answer your question?
Yeah, it was cool. Thank
Okay.
you.
I lost the thread a little bit at the end.
Hi, thanks so much for your talk, Rachel. I was curious, in terms of the intertidal zone situation, I'm actually curious whether the number of hours that a mussel is not covered by seawater might correlate to morphology in some way, and whether or not it's thicker, able to repair as quickly, for instance. If it's able to filter feed and get more energy. Just curious about that. But I had a couple questions before that came into my mind.
Yeah.
One was, is there a difference between the density of a shell that's been repaired than a part of a shell that has not been repaired? And then my other question was, are there differences in the work that you've done looking at morphology in the higher and lower intertidal? Mussels are usually in clumps, and so the ones in the middle are kind of protected against most of these kinds of damage, I would assume, either predation or getting hit with a big rock. So I'm curious whether there are differences in the middle of the pack, and if there's kind of a selective advantage to being in the middle, or do you get stronger when you're on the outside?
Yeah.
I think it'd be interesting to look at that in one of the other studies that you didn't get a chance to do.
Yeah. Variation within the bed is super interesting. I have no idea about that. And then, yeah, because you could imagine both sort of relevance of predators and kind of random debris hitting from the sides might be higher at the edges, but I don't know how any sort of, you might get more wear in the center. I don't know. It's interesting. Yeah, and then, okay, that was question 2, which I don't have any direct thoughts on, except like hadn't thought about it. It's super interesting. Can you remind, there was a question in the middle?
Density.
Density, yes. Okay. So I started some of this work, didn't finish or publish it. COVID interrupted a lot of things, but I think I saw someone published on this pretty recently looking at the microstructure. So I don't know specifically about the density, but the microstructure of those patches is really different. So the crystals, right? It's getting the nacre, the calcium carbonate. So the normal internal layer, the calcium carbonate crystals are in bricks, like very organized as they're laid down. And the I don't know if I have any pictures in here. The repair patches almost look like they're way more chaotic, and I do have pictures. Wow. Okay. So, okay. So this is the nacre, and you can see, right, like here's like these beautiful bricks. They're all neatly stacked on top of each other. This is more zoomed out, and you can see neat bricks all on top of each other. Here is a patch. So here's our beautiful nacre bricks, and then here's this like big, messy shingles of calcium carbonate crystals. Sometimes they were organized more haphazardly, but they were sort of tended to have a direction to them. This is a little bit more zoomed in. I really am very curious about how the mechanical properties of these two things differ. Building nacre is well known to be pretty costly. It has a higher amount of organic content, and it has a lot of really amazing crack arresting abilities. But I'm really curious if there's some trade-off. Like, is this faster to lay down but worse? Or is this great? I don't know, but I think that's really interesting. But it's not the same thing. It's not recapitulating the same normal internal structure.
We have a question from online. Sorry, Allison, I see you. This is from Maricela. Thanks for joining, Maricela. So Maricela says, super interesting to think about fatigue in shells. What led you to study this, and what kinds of questions are you hoping to answer next? And has there been any interest from biomimicry or industry in this sort of work that you're doing and discoveries you're making?
Okay. Okay, so what led me here? The work, it's funny, it felt like I was making a big transition, but it's so linearly coming from what I did before this. So I worked as a lab manager for a few years before graduate school, studying mantis shrimp and looking at mantis shrimp that hammer open hard-shelled prey. And I was doing this behavioral study about sort of it turns out they are targeting specific parts of the shell when they hit them, and that what part of the shell they target changes depending on the shell morphology. And also, like, I feel like, for people who don't know, mantis shrimp, they're these really amazing marine crustaceans that have this really, really cool, highly modified mouth part that uses a spring to like swing this hammer forward that accelerates like a bullet in the muzzle of a gun. So it's this really, really fast system that's able to shatter shells, and the story is always about how powerful these are and how much damage they're able to cause. But it turns out, like, you give a mantis shrimp a shell, it's like 5 to like hundreds of strikes later before they actually crack open the shell. Like, they really, really have to work at it, and they're hammering away. So fatigue, like, really direct line between these ideas. That's how I got started thinking about fatigue. One of the things that I would love, we talked a little bit about, I really would love to do this kind of work more broadly across, someone already asked about this, across different kinds of shells, and seeing how this relationship changes depending on the ecosystem and the life history of the shell. And then, there's like the even farther down the line, you could begin to think of, if you do that broadly enough and then look across the amazingly rich paleontological record for these shells, looking at evolution of shell form and how that relates to different ecosystems and amount of predators and the demands on those shells. Was there another question?
So the second part of that question was just if any industry, if there was any interest like biomimicry industry or any applications in industry that you think this could apply to.
Yeah, so there are a few sort of like in the world of biomechanics, right? I'm like way off on the like I'm a biologist who dabbles in some fun tools, and then there's a set of like really intense biomaterials, really impressive people who are Laura's laughing at me. Thank you. Who just have a very different skill set from me, who are doing like really in the weeds materials work, especially on nacre, about sort of all of its amazing properties from a directly biomimetic perspective. But I do think, I think there's a lot that could be learned from looking holistically at the whole shell, because that functions from all of these layers working together and not just from one layer isolated on its own. Oh, but yes, making more fatigue resistant ceramics is really important, and manufacturing ceramics, shell is a type of ceramic, it is really hard to do the types of things that these animals are doing just naturally without thinking about it. So yes.
You've already answered a lot of questions, so I won't hit you with too many more, but I do have a lot. One is, okay, so I don't know if I missed this or if this was just something that's maybe not that important, but when you were showing your plots from your first set of questions, or your first part of the talk, you talked about predictive strength.
Oh, yeah.
How do you calculate that, or how do you come to, because that's like really important for all of your trends, because it's the y-axis, right? But it's predictive. So how are you measuring that or estimating?
Yeah, this is kind of fun, and I cut it out for time, but I think it's pretty clever. So thanks for asking about it. So bivalves have two valves. It's like magical, right? Each shell comes with its own little buddy counterpart who experienced a lot of the same life history as it did. So for every point on this graph, this is one mussel valve. The valve that was its partner valve, I strength tested that valve. And then I used that as a baseline, and I adjusted it based on morphological differences between the two valves. So yeah, that's how I calculated predicted strength.
Okay, and then my next question is that one guy that lasted over a year. Like, why do you think it was so strong?
I mean, but it's not actually, right? Like, it is smack dab in this line, right? Like, it is lasted for 409 days because I was loading it at like 20% of its predicted strength. So it's funny, right? Like, these are like biomaterials. This type of data is by definition really noisy, but this shell, it's like hard to imagine it being more smacked up on the line. But the reason it lasted that long is just that it was loaded so little that it was able to hold out for that long.
But its predictive strength was already really low because it wasn't like its buddy shell wasn't that strong to start with in this case, right?
Well, so the buddy shell was exactly as strong as, I actually have no idea. I don't know for this shell. I think it doesn't matter how strong the buddy shell is. What matters is that this shell was loaded a small amount relative to that buddy shell. So if that buddy shell was 100 newtons strong, this shell was loaded at 20 newtons. So it was just a really, really low force that I was applying here. Okay.
Yeah. But you also did that for other shells that still didn't last that long.
Right? Yeah, right? Like this guy, I don't know what happened here. Like this shell was not very good for sure. And I mean, yeah, sometimes shells have like obvious pre-existing damage and they break right away, but then it's also it's really variable, right? These animals they're depositing their own shell, weird things happen. There's all sorts of things that I don't know about their history. And any sort of biomaterials testing is by definition really, really noisy. Often when people are doing this sort of mechanical testing, they'll work with strips machined out of a shell. So like you'll take a shell and you'll cut a strip that's like 2 millimeters by 4 millimeters. That's really small. 1 centimeter by 2 centimeters from a certain part of the shell. And you'll do that for a whole bunch of shells, and then you'll test that one little piece of shell. And that tends to give you much cleaner data. But I think it's much harder to extrapolate from what does the data from that little piece of shell tell you about how the whole animal interacts. So there's a trade-off where these data are noisy because there's a whole structure there that some animals build bad shells, or maybe yesterday a crab tried to crush them and they survived. Like, I just don't know. Yeah.
Okay, I have a lot more, but can I ask one more? I finally have the microphone. So what do you think it is about the lab conditions that you're missing? Because we work with lab animals too, and it's something that like we struggle with, and just trying to figure out like how to get lab critters up to speed with the natural environment.
Yeah. So I had nothing nearly as fancy as y'all's like beautiful and expertly maintained systems. And this was also like me during COVID trying to take care of these animals. I did, the system was programmed. It had a tidal cycle. The water rose and fell. But I suspect I was not feeding them enough. My understanding is that mussels in captivity really, because they're normally filter feeding anytime the tide is high. And we did not have an automatic feeder. So I had to be there in person to be feeding them when they were underwater. And I think that I just couldn't do enough of that. That's my best guess. But it could be, I mean, they were in a flow through system, like, so it was their water, but it was also filtered. And, they're not getting their normal stimuli. They're in a little pavilion. So I don't know. It's really hard.
We have one more actually from Marcella online.
Okay.
And it's just wondering, bringing it back to museum setting, if and how preserved collections might support your work.
Yeah, I think there's a few. So I think looking at, getting like opportunities to see how patterns in changes in shell morphology across time, or the sort of either geographic or spatial scale. Wait, those are the same. Geographic or temporal scales that can be preserved in museum collections. And then I don't know, I should talk to you museum experts about this, about like how, I don't know how much there is. Yes, I think changes in shell morphology are the most obvious, but I also don't know how much you can learn from the like how often there are fractured shells, if there are interesting things in shell fracture patterns that might be preserved. I don't know. Obviously, smashing all the fossils are not, smashing the museum collections is both, I'm going to tell you right now, it is both not an option and also is not very informative. Like the living parts of the shell and the organic components are really important. So there's, as much fun as it would be to destroy these amazing collections. Nope, don't want to do it, wouldn't learn anything from it. Just trying to run with, I also don't know how, like, I guess if we're looking at morphology, I think microstructural patterns could be really interesting too, doing SEM work on them.
Yeah.
That's what I got.
Absolutely. Thank you. Any other questions from the room? We've peppered poor Rachel with enough questions.
Well, thank you so much, everyone, for joining us and for hosting Rachel with me. And thank you, Rachel, for a wonderful talk. And she'll be at IVSS Cheers if you want to pepper her with more questions in just a little bit. Thanks.